Let me start with saying that I'm really deeply honored by receiving this award, and I'm also very proud of getting this award. So I'm very grateful to EBSA and Avanti for awarding me this. What I'd like to do today is talk about our work on protein-lipid interactions, going from model peptides in membranes to nanodiscs. And this looks like very different things, but in the end I hope that you realize that they are can be very closely connected. Let me go back in time a little bit. I've been working all my life, my scientific life, that is, on lipids and lipid-protein interactions. And the very first time when I encountered lipids was when I went to the lab of Laurens van Denen in the group of Hans de Gier and Ben de Kruijf. Difficult names, I know. And I had no clue at that time how famous they were and how famous that lab was, but they were working on these model systems. And I was very much intrigued by these model systems because by using only one single type of lipid and an aqueous solution, you could get vesicles. And with these simple systems, you could do many different things. You could look at leakage, so permeability experiments. You could look at the rich phase behavior of the lipids. And you could look at binding of all kinds of things. And this all gave you information about what could be happening in real biological membranes. Now, I'm still excited after all these years about what you can do with model systems. And I hope that after this talk, you will understand why I'm so enthusiastic about them. So, protein-lipid interactions. Before I start, I want to stress two things. One is that membrane proteins, I think all of you know this, they are abundant. They have many important functions, but they're very difficult to study. And that is because they're hydrophobic, they sit in a membrane, so they have hydrophobic amino acids here, and that makes them get a tendency to aggregate as soon as you want to purify them. The second thing I want to stress is that lipids are important for this membrane, for the structure and the function of these membrane proteins. You can imagine, for example, if this is the bilayer, it's very hydrophobic. If the length of the lipids would be shorter or longer, this would affect the conformation and the behavior of the protein in the bilayer. There could also be electrostatic interactions, or a lot of those proteins, they have to move while they function. So the packing of the lipids around such a protein is very important to allow the protein to move. So if you want to study proteins and how they are influenced by lipids, then there's several ways to go about it. But the simplest way to do it is the following. You just take a single transmembrane helix as a model for a membrane protein. You try to understand everything about how this single transmembrane helix affects the lipids and how the lipids affect this single transmembrane helix. And then from there on, you start a build. And this is what we try to do. And I will give you some examples of how we approach this. Well, this looks quite easy, but it's not so simple as it looks, because the helix that you then design, it has to mimic the transmembrane segment of a real membrane protein. But on the other hand, it should also allow systematic variation of the properties of this helix, because only then you can do systematic studies and start understanding things. Now, for the design of this helix, I want to go back a little bit in time, back to 1992. And that is the year that Roger Cuppy visited our laboratory on a sabbatical. And Roger was working on grammacidin. And at the time, I also was working on grammacidin. And I was working on grammacidin lipid interactions, and Roger was working more on the channel-forming properties of grammacidin and on synthesis of all kinds of grammacidin derivatives. Now, grammacidin is a very special molecule. It's a cation channel. it has a very strange structure of alternating L and D amino acids, and that makes that the peptide forms a beta helix. Now, it can form all different types of beta helices. It can form an anti-parallel helix, but it also can form a helix or a double helix with two N-termini. And this seems to be the preferred orientation in the membrane. And that's amazing because these two structures, they both have approximately the same dimensions. They both have the hydrophobic amino acids pointing outwards, and they both would be able to form this channel. So why would this one be preferred in bilayers? Now, at that time, from our own studies, it became clear that these tryptophans here were very important for establishing this conformation. At the same time, it became also clear from the work of Steve White mainly that tryptophans have a very strong preference for the interface. So the hypothesis then was that because the tryptophans here are all near the C-terminal end of this molecule, that it was the tryptophans that made the interaction of the tryptophans with the interface make this conformation more favorable. Now, around the time, it also became more and more clear that the membrane protein structures that were solved, that they all contained tryptophan or other aromatic amino acids near the lipid-water interface. So this sort of then, so Roger and I wanted to make an alpha helical peptide that we could use for systematic studies. And with all these considerations, we came to the following. So it's a sort of alpha helical derivative of grammacidin that allows systematic studies. So this was our peptide that we designed. We call it WALP because it has tryptophan, alanine, leucine. It's tryptophan, alanine, leucine peptide. It has two tryptophans on each side, at the N-terminus and the C-terminus. Then a stretch of alternating leucine and alanine. And you can systematically vary the length here. The N and the C-terminus, like in grammacidin, are blocked, so there are no charges, so we don't have to worry about electrostatic interactions. So this was a very simple model system just to start with. WALP 23 has 23 amino acids in total, and that's all the time the notation. So it's simple, uncharged, and it mimics a real membrane protein in the sense that it's alpha helical forming. Leucine and alanine are both amino acids with a high propensity for alpha helix. They have average hydrophobicity, and we have tryptophans at the interface. Because you synthesize these peptides, you can also incorporate all kinds of labels that you want. And so this allows very systematic studies, because you can vary the length, you can vary the flanking residues if you want to know why tryptophans are important. You can vary the hydrophobicity, you can vary the helicity, you can incorporate prolines, glycines, you can do whatever. But this is the one we started with. Now you can vary all kinds of things. We decided to focus on hydrophobic mismatch. And why? Two reasons. Because it occurs in biological membranes. So not all transmembrane proteins have the same length. Not all membranes have the same thickness. And some proteins, they have to distribute between different bilayers. So it is encountered in biological membranes. And the interesting thing is, if there is a mismatch, it can have many potential consequences. Think of what could happen if the length of this, the hydrophobic length of this peptide is either larger or smaller than the hydrophobic thickness of the bilayer. These are just, there are many things that can happen, and these are just a few. Suppose the peptide is too long. What could happen is it could start tilting, or it could deform, or get another kink, or get another conformation, or it could start aggregating. But also the lipids could adapt. They could stretch if the peptide is too long, or they could deform, disorder if the peptide is too short, or at high concentrations they could even form completely different lipid phases. So it seemed like already a big challenge to find out how such a simple peptide would affect or would be influenced by the lipids and how the lipids would influence the peptide. So the basic approach is then the following. Very simple. We use peptides with varying length. We vary the thickness of the membranes, and then we analyze the consequences with different biophysical techniques. And I must say that one of the great advantages of using this system was the fact that there are so many different lipids available from this company, Avanti. And they have all different length and different saturations, but also with different labels that you can use for biophysical studies. There's not enough time to show you exactly all the data, what we found and how we did it. So what I do is I will show you the outcome, and then I will zoom in specifically on what happened to the lipids. So first, the outcome. We found that several adaptations occurred at the same time. It was not one of them, but several. So what happened? The peptides were tilting more if we decreased the bilayer thickness. So if there was more mismatch, they tilted more, but just a little bit more. Very small effects, but very systematic. This did not happen. They formed very stable helices. Aggregation also occurred, both with negative and positive mismatch. The lipids also adapted, just a little bit, but very systematically, and also with negative mismatch. Now, does it make sense? Yes, I think it does, because you can imagine that a little bit stretching of the lipids is energetically still favorable, but the more they need to stretch, the more unfavorable it is. The same for tilting. A little bit tilting is okay, but the more they tilt, the more they disorder the lipids, the more difficult it gets for the peptide to rotate around its axis, to reorient about its axis. So apparently the system chooses for several ways parallel to deal with this mismatch. Now let's zoom in on this effect first. So the stretching of the lipids. How did we do it, and what can we learn from the experiments we did? Now these were experiments done by Maurits de Planck already quite some time ago. And what he did was using deuterium NMR. We used per-deuterated lipids, in this case the very short ones, C12. And then he incorporated peptides with different length. 16 amino acids in total, 17 and 19. So it was just the leucine alanine stretch which was becoming larger. And what you see here is that the distance between these two peaks is increasing. And that is a measure of the order of the lipids. It means the lipids are stretching. And you can estimate how much they are stretching. And then you see that it's very systematic. A short peptide stretches only a little bit. If you increase the length of the peptide, the lipids stretch a bit more. But what you can also see is that the difference in length between WALP 16 and the WALP 19 is 3 amino acids. There will be 4.5 angstrom. And the difference here between 0.6 and 1.4 is much less than that. So there's only a little bit of stretching, but it's there and it's very systematic. Now, what can we learn from this? For example, you want to know the tryptophans, they are at the interface, they have a strong interaction with the interface. Is it the tryptophans that are important for this stretching? So if you want to know this, what do? You can replace the tryptophans by lysines, for example. So instead of a WALP peptide with the tryptophans, we now have a KALP peptide with lysines. does such a peptide have the same stretching effect? Now you can do the same experiment again. And here you see that even though the peptides are a little bit longer, and this is just an example, Maurits did all different lipids and all different peptides, that there was maybe a little effect, but it was very, very small, almost negligible as compared to these tryptophan flank peptides. So this, I think, clearly shows that tryptophan flank peptides, they can stretch the bilayer, so supporting a very strong interaction of the tryptophans with the interface, and the lysine flank peptides do not. And now let me go to the other situation with a negative mismatch. If the peptide is too short, I will focus on what we saw with the phase behavior, because that gives the most exciting results and the most clear results. So there was a systematic effect here with the deuterium NMR at low concentrations, but at high peptide concentrations you can see really changes in phase behavior. Now before I can explain this, I have to make a short detour towards phase behavior of lipids. So lipids, they have a very rich phase behavior. And normally lipids that have like a cylindrical shape, like a phosphatidylcholine, they like to form, organize in bilayers. That packs very nicely together. If lipids have a relatively short small head group, or a relatively large area of the acyl chains, then they like to form curved structures. Now, if this curvature is not too much, like you have just sort of intermediate, then the systems often tend to form cubic phases. But if this curvature is getting more, and if it's really highly curved, like for a phosphatidylethanolamin, then the lipids tend to form a hexagonal phase. And this is a phase in which the lipids organize in long tubes. There's water inside the tubes, and the head groups of the lipids are pointing towards the inside of the tubes, and you have 6 tubes surrounding one tube, and that's why it's called a hexagonal phase. Now, if you think of this picture, and now you think of a peptide in a lipid bilayer, then you may expect something similar happening. Suppose this is the peptide, this is the position of the tryptophan. Suppose the tryptophan likes to anchor at this position. Then you make the peptide shorter. If there's a strong interaction with the lipids, then it may induce a curvature. And then if you make it even more short, then the curvature gets even stronger. So what may happen is that with mismatch you go from bilayer to cubic to inverted. And this was indeed what happened. And this you can see from the NMR spectra here. Now, even if you don't have any idea what the spectra mean, you can still see a few things. You can see that if you have a phosphatidylcholine with different length, they all have exactly the same spectrum, representative for a bilayer. And also, if you incorporate these peptides, WALP 17 and 19, in the shortest bilayer, you have in the shortest bilayer, you have a bilayer spectrum. Then if you have the WALP 17 and you increase the thickness of the bilayer, then something happens. You get an isotropic phase, which is characteristic in this case for a cubic phase. If you increase the length of the peptide, then in the same lipid you don't see anything happening. But if you then also increase the thickness of the lipid, you see something similar as here. Now, and this is interesting because the difference between those two amino acids is about 3 angstrom, and the difference in thickness between those two is also 3 angstrom. So it means that this is really a mismatch effect. If you have longer lipids, longer peptides, you need longer lipids. If you go to even longer lipids, then you see this one is disappearing again, and you see something coming up at a little bit lower field. And this is characteristic for an hexagonal phase. And this is the same that you see when you go to a longer peptide and a longer lipid. So it's very, very systematic. Now we did this not only for these, but many other peptide lipid mixtures. And these are just all the type of samples we took. So we calculated the mismatch just from the length of the peptides between the tryptophans and the distance of the bilayer between the carbonyls. And then if there's a positive mismatch, then we found a bilayer spectrum. An isotropic phase was occurring at a certain quite sharp boundary, and here again at a quite sharp boundary we found the hexagonal phase. Now the reason why this is so interesting is because of the implication. Because it means that this phase behavior of the lipids can be used as a ruler, a molecular ruler for the effective length of for the extent of matching, and so for the effective length of peptides. Okay, so for the tryptophans this worked. Now, and this suggests also, is again support that the tryptophans really interact with the interface very strongly. What happens now if we replace the tryptophans by lysine? Now lysine is a bit different in that the tryptophans are supposed to anchor at this position, but the lysines more around the phosphate. So you would expect that a matching length of a lysine flank peptide would be a bit larger. And you can calculate, so in a bilayer, this difference here would be 8 angstrom. So what you would expect is that a lysine flank peptide with 23 amino acids should behave about similar as a tryptophan flank peptides with about 18 amino acids. This did not happen because what we found was that it behaved like a much longer peptide. And so we thought, what would this mean? And we came up with the following, that apparently, this suggested that the PC system that we're using, the phosphatidylcholine, it doesn't like to form these hexagonal phases. It's a real bilayer lipid. And what is happening then with the peptide is that when it gets shorter, the system doesn't really want to go into this hexagonal phase, so the lysines, they start snorkeling. So the peptide is making itself as long as possible, sticking the charges out. And Erik Strandberg in the group, and he's here in the audience also, he then did experiments putting the same peptides in lipids that by themselves have a tendency to go to this hexagonal phase, and there indeed he did not see the snorkeling anymore. And this gave us an idea about the snorkeling. So by doing these studies, we did get information about how amino acids behave at the interface. And at about the same time, Gunnar van Heijnen in Sweden, he was doing experiments with this glycosyl transferase that he talked about in his opening lecture. And doing this assay, he also got information about the behavior of amino acids at the interface. And this correlated really well with our studies. So we decided to write this small review together. And the cartoonist from TIPS, he apparently was inspired by our paper because he was drawing this cartoon. There you see the alpha helix crossing a lipid bilayer, and here you see the tryptophan very firmly anchored at the interface. Here's the lysine snorkeling, and also the arginine can do a bit of a snorkeling. So then, with these peptides, we can get information about what is happening as function of mismatch. How do you continue with this kind of experiments? There's several ways for following up. You can, of course, then systematically start varying other parameters, like putting in prolines or changing to other amino acids or making it more hydrophobic, and try to understand even more about peptide-lipid interactions. You can make it more complex, make mixtures of membranes, look at whether the length is important for sorting, what properties of the peptide and the lipids are important for sorting, whether peptides can attract the right lipids around them, matching lipids around them. You can also make the peptides more like larger membrane proteins with multi-spanning domains, but still in a way that you can very systematically vary them and then see what the size is doing to the protein-lipid interactions. And what you can also do, and this looks very far away from this one, is looking at native nanodiscs. And I now want to continue with this is our recent research on these discs. And before I can explain this, I need to give a little bit of background. And that is, so if you want to investigate protein-lipid interactions of native membrane proteins, what you normally do is you have here the native membrane, then you have to isolate the proteins from the membrane, and that you normally do by using detergent. And then if you want to know how they interact with lipids, then you can reconstitute them in membranes or vesicles, and or you can reconstitute them in nanodiscs with a certain lipid composition. Now these nanodiscs here, they're protein-stabilized nanodiscs. They were developed in the lab of Steve Slieger a number of years ago, and they've been very popular because they have a number of advantages over these vesicles. Of course, the vesicles have the advantage that you can use transport, that you can do transport studies, but the nanodiscs also have several advantages. They're very small, so they're easy to study, much more easy. Both ends are accessible to the aqueous phase. And also an important thing is that once you have captured a protein in a nanodisc, it has a defined oligomerization state, and this oligomerization state will remain. Now, a very recent development is that the use of a polymer, styrene maleic acid, and this polymer is able to directly cut out nanodiscs from native membranes. And this has a tremendous impact, because if you would have a protein with a his tag, then you can just add this polymer, you get the protein solubilized in its native environment, and you can purify the protein just like you're used to purifying a detergent solubilized protein, or almost similar. So what does this magic polymer look like? It's this one, alternating, or not alternating, but consisting of styrene moieties and maleic acid in different ratios. Yeah, you can get them in different ratios, just changing the hydrophobicity. Now, this was first published in 2009 by the group of Mike Overduin and Tim de Voorn. So they published this method, and it looked very promising. And then very soon after, there came a lot more studies from the group of Tim de Voorn to characterize these nanodiscs. And also there came a number of studies of the group of Tony Watts, characterizing these nanodiscs and applying them to different membrane proteins. So it looked really promising, and more groups, I think, have started working on them, on these polymers. And there are several posters here now, and this afternoon there's also a talk about the use of those polymers on membrane proteins. So they're really promising systems. And we also started working on them a few years ago, and on the reaction center on KCSA, and we were really excited about how well they worked. And I want to give you an example now of what we did on those with those polymers, and that is the work of Jonas. He is here in the audience, and he's presenting this poster. And he worked on KCSA, so the protein you've seen in featuring in in a lot of previous cartoons, except this is only the transmembrane segment, and this is a model for the full length. We like the idea of working on case-TSA, of trying this with case-TSA, because it's a very stable protein. It's a complex protein, not really. It's a rather complex protein because it's tetrameric, but it's very stable. And the most important thing is that it has lipid-dependent properties. So it needs anionic lipids, for example, for functioning. Now, Jonas showed, and you can see it on his poster, that you can just grow E. coli's with a case-TSA with a his tag, add this polymer, and then purify it, just like when you would have added detergent. Now, what can you do with those purified case-TSA nanodisc? Well, remember, they retain the native environment, so you can biochemically analyze them. And that's one of the things that Jonas did. He just looked at the head groups of these nanodiscs and compared them with the total lipids in the bacteria and the total solubilized lipids, and these are the ones in the nanodiscs. And what he saw that in the nanodiscs with case-TSA, there was an enrichment of cardiolipin, PG, and these are the negatively charged lipids. So that was sort of kind of nice fitting with what is known already, that case-TSA has a preference for anionic lipids. He also looked at fatty acid analysis, and there it seemed there was not a big preference, except there may have been a small preference for the longer versus the shorter lipids, but I think it needs more measurements to say something about this. So that's one thing. You can look at the lipids, at the native lipids that were surrounding the protein in its native membrane. Another thing you can do, and that is because the polymer is not active in CD, it's not a protein, is that you can do CD. So you can directly take a CD spectrum of the protein in the nanodiscs, and this protein is still in its native environment and has never seen any detergent. What you see here is that at 20 degrees, you have the same alpha helical spectrum for DDM and in the nanodiscs. But if you start heating up the sample, then even to 95 degrees, very high temperature, that there's still a considerable amount of alpha helical structure left when you heat up the nanodiscs. And in DDM, it's losing its structure. So these nanodiscs are really stabilizing quite well. But maybe the most spectacular thing that Jonas did was the following. He added those nanodiscs, because you cannot look at transport function in the nanodiscs. He added them to a black lipid membrane system consisting of E. coli lipids, and then he found that you get conductivity traces that are or they're equivalent to those of case-TSA. And this suggests, or this indicates, that the protein is able to transfer spontaneously from those nanodiscs, apparently in this bilayer. Now, another thing we then wanted to do is we wanted to know how does the case-TSA work. And here are the model systems again coming into play. This is work by Martijn and Stefan in the group. The idea was you start with vesicles, and they're rather large, so they are turbid. Then you can add the polymer, and just by looking at the optical density, you can follow the formation of those nanodiscs. And it's a very simple assay, but it allows you to look at all kind of different parameters. You can look at pH, salt concentration, different types of lipids. And this is one example of the type of information you get. So this is solubilization of a DMPC, which has a transition temperature around 23 degrees. Then you can see that below the phase transition it goes slowly, but at and above it goes very fast. Now with using a lot of different variables, we came to this model that a lot of parameters are important for the efficiency of binding. So it goes a bit less if you have anionic lipids. If you have salt, it goes better. And of course, with more SMA, it goes better. Lipid packing and bilayer thickness are very important for the extent to which it can insert and the efficiency of the nanodisc formation. And this way, you can very systematically find out more about the process. Now, I'd like to end with some possibilities and challenges of the system. When you have a protein in a nanodisc, you can, of course, and many groups are now working on this, trying to use it for many different proteins, many different organisms, and I think with quite some success. So I think we'll read a lot more in literature about this very soon, and papers are already coming up very fast. Of course, they are very promising also for characterizing protein structure with EM, like you may hear in the talk this afternoon, solid-state NMR, maybe mass spec. You can use them, like Jonas did, for protein stability, assembly, function, and of course, characterizing native interactions, like looking at the lipid interactions. That's one possibility. The second one would be, and that's a real challenge still, going from nanodiscs to membrane vesicles, or to real membranes. Why would you want to do that? Well, for example, if you want to look at transport properties, but also if you want to have the protein in a well-defined lipid environment. You could also then, if you have it in a lipid environment, add a SMA and get back to this nanodisc, and that would allow you to get a protein in a well-defined environment. And other challenges are, of course, to replace the polymer by this protein, because the polymer has a lot of advantages, but also some disadvantages, like it's highly negatively charged, and this one is better defined, it's more functionalized. You can get it into bicels, and maybe this will help you to get it in a lipidic phase for EM, for crystallization studies. And now I'm coming back to the WALP and KALP peptides, because I think also they're very promising for really systematic studies. You can put them in, these peptides, in the membranes. Suppose you take mixed membranes of like 2 different thicknesses, and then you can add a SMA, isolate the particles with the peptide in there, and see if they have attracted specific lipids around them. So you can look at preferential interactions. You can also look at aggregation. You just add the SMA again, vary the protein-lipid ratio here, and then analyze the protein-lipids ratio in your particles and see if they have aggregated. So these are lots of things you can do with them, and there's much more, like functionalizing the polymer, or finding out more about the energetics, thermodynamic parameters. Okay, so I'd like to leave it at here, but I would like to thank a lot of people. First, our group in Utrecht, membrane biochemistry and biophysics. A lot of hardworking and enthusiastic young people, and the people within the group that have been working on these, on the model peptides. They're all listed here. A special thanks here for Ben de Kruijf, who taught me all about, or a lot about lipids. These are the people working on the small ping. And finally, also a lot of collaborations outside the group. Again, on the model peptides, on the small on a small project, and there's also a lot listed of collaborations that have just initiated, and I'm also grateful for those collaborators. Here there are two people in red, Roger Kuppy and Jeroen Lindblom, and I would like to thank them especially for long-standing and very special collaboration, and all these for funding of these projects, and Polyscope for the collaboration in the project with the nanodiscs. And I would like to thank you for your attention.
All right. So it's a great pleasure, Antoinette, first of all, to listen to the wonderful review and perspective into the future, but also to hand you over in my new capacity. That's the first thing I am allowed to do, which is wonderful. There are worse things to do. There is a document with it. Mm-hmm. It says Antoinette Killian.
That's me.
And the problem with these documents is, unless you keep them in the fridge, they may disintegrate. And so probably that is
should
number
make a picture. Okay.
But we also thought we'd give you something more durable, which you don't need to keep in the fridge. You know, you've got all this stuff in the fridge which
I'm not
might
planning to
spoil
keep this
that
in the
one.
fridge.
And this is a medal, and I really liked it very much. It says it's the Avanti Lipid Polar Lipids Award, and it's a great thing. And
that's really beautiful.
It's a pleasure to hand you over that. Very well earned. Mm-hmm. And now I'm rid of it, and you have to carry everything.
Okay.
All
Thank you very much.